Well, it's a great pleasure to open this meeting. I'm glad to hear that there's almost nothing been done on this topic I've been working on for the last 10 years. But we'll see how far we've got. Sorry, I'm pulling your leg. So my background is studying evolutionary biology, and for my whole career I've been interested in the question of why one organism would cooperate with another in particular, or indeed compete. And by cooperation I mean sort of a phenotype or behavior in animals in one individual that has a benefit, a fitness benefit to another. And this was one of those things that Darwin was very interested in. He didn't see a huge problem with it, but he recognized that if you could show that one individual was helping another at no sort of benefit to itself, then we had a problem for his idea. And since then there's been a lot of work on these things, and I think in animals we certainly have a pretty good idea of how and why they cooperate. But then the last 10, 15 years or so, people, including myself, have got very interested in the same question in a different group of organisms, and that, of course, is the microbes. And we now know that microbes very commonly live in dense and genetically diverse communities, where they meet both with sort of family members, if you like, with clone mates, but they also meet with many other different genotypes, both different strains and species. We also know that in these communities, their ability to grow and divide and prosper often rests on things that act outside the cell. They secrete many enzymes that help them to grow. They secrete quorum-sensing molecules, a form of communication. And they also secrete things like antibiotics, which have very negative effects on their neighbors. So we know that in these dense communities, there's many genotypes interacting, there's many different traits affecting those interactions, and that the sum of these things are critical to how microbes function as a group, and indeed how they affect us. But as we've just heard, this is a complicated problem, and so the question is, can we try and dissect it and understand sort of the rules of interactions in these communities? And that's very much what myself and several others have been trying to do. So sort of the leading question for the whole of my talk is, what do microbes do in their communities? Do they tend to invest in sort of goods, if you like, in positive interactions where they help cells around them, or do they tend to invest in negative ones? And indeed, what determines whether they do one or the other? So to answer this, we can sort of borrow very heavily from sort of the core principles that were developed first from animal behavior. And I'm just going to translate sort of one of the key arguments in evolutionary biology into microbes, and this is a cartoon I show at the beginning of all my talks. So what we're going to do then is we're going to do a little experiment where we take a secretor cell, and the secretor cell is going to release something that helps everyone around it to grow, but we're going to mix it up with a non-secretor cell. So this cell, will say it's isogenic, but it lacks a particular, the particular enzyme, say, that breaks down a resource in the environment. And we're going to put these under two conditions, but let's start with the top one. Under well-mixed conditions, we allow our strain to secrete its factor, and it helps the cells around it to grow, but it's only the one that's actually paying a cost. So under these conditions, we might expect it to do rather poorly, because what it's doing is it's releasing something that helps everyone to grow, but it's the only one that has a cost. By contrast, we might do one manipulation. Let's put them in a little clump. And in doing so, we might expect something rather different. So now in our little clump, our red cells are preferentially helping their own genotype, and we might expect something rather different to happen. Now they can prosper, because even though they're paying the cost, they're also the only one getting a benefit. So this then recapitulates one of the sort of key arguments, as I said, in evolutionary biology, which is if you're surrounded by sort of family members, then you can invest in your own genotype, and you can get what we call cooperation evolving. And so that sets up basically a prediction. Under conditions when things are very mixed, we might expect the cells actually they don't invest in this enzyme. They might instead invest in an antibiotic. By contrast, under conditions where they're clustered, we might expect them to invest in a cooperative trait. And because this, the sort of title of this symposium was very much sort of based on communication, I would like to also translate the predictions in terms of communication. Well, in evolutionary biology, we take communication to mean where two individuals are actually sort of signaling to each other cooperatively where they both benefit. So like two people talking together. And so the bottom case is where we expect things like communication to evolve, and that would be things like quorum sensing and things within a genotype. By contrast, up in our competitive arena, we expect it more to be about eavesdropping, so listening in on your competitors to get an advantage, or indeed maybe manipulation, so secreting something to induce something in them that they actually don't want to do. So What evolutionary biology says then is, communication predominantly we should see in the bottom case. In the top, we should see sort of these manipulative interactions. Okay. So my talk has three parts, but the first part I'd just like to focus then on these clonal groups and what we can get out of those. And as I said, the prediction is that living in a clonal group is a place where we can expect to get cooperation. But the key corollary of that is, when do we see that? How often do microbes actually exist in these little clonal patches? And this is something we've been working on for a while, including with João Xavier, who's here, he'll be speaking later. And he and I, several years ago now, started just to play around with both an individual-based model and then ultimately experiments to try and understand under what conditions do you get clonal clusters within microbial groups. And this is relatively old work now, so I'm just going to sort of run to the punchline. But if you this is on the left is a simulation model where the two genotypes are just identical except for colors. But what we have in this model are diffusion reaction calculations that mean that the cells actually are using up virtual food, and the cells on the outside take food and starve the guys on the inside, as you commonly see in microbial groups. And as what you saw happened when we grew them, they started off well mixed, but by the end they're demixed, right? They look what I like to call psychedelic broccoli. But the key point is that each floret of our broccoli is a different color. So they've demixed and they've ended up in these patches. And this happens actually spontaneously. The reason is just due to the fact that only the cells on the outside of the group are growing well because there's nutrient limitation. And so you get stochastic events and you end up basically with pure patches of a single genotype. And we can run the same experiment in a real case. This is Pseudomonas aeruginosa, that's a well-studied pathogen and a focus of my own lab. And all we've done here is mix up two different colors of a single strain background, so identical except for the color. And we can mix them up, put them down in a regular colony. So this is just a regular colony, but imaged with fluorescence imaging. And you can see the same things happening, where over time you get basically statistical sampling at the edge of the colony. Only certain cells get to grow because there's limited resources, and you end up basically with these large clonal patches. So under many conditions then, microbes spontaneously sort of solve this problem of being surrounded by their own genotype just because of the way they grow. So that's quite good. That suggests then, well, maybe at the scale of these patches then, we can expect there to be the evolution stably of cooperative traits. Well, let's test that. Well, we can test it first in a simulation model. So we're going to run a model here again, and we're going to have a secretor and non-secretor cell, as in my original cartoon, but I've just changed the colors to make it confusing. And in this case, the secretor cell is a blue guy, and the blue cells will secrete a factor that in the area around them will help everyone to grow, but pay a strong cost for this. The non-secretor doesn't pay the cost, but doesn't make the product. If we grew these two together under well-mixed conditions, what we see is rapidly the non-secretor cell will take over. But if you put them into these sort of nutrient-limited conditions where you get bottlenecks, you see the opposite. So now you get these sort of, you get basically statistical sampling again, so only certain cells can grow at the very edge. That creates little patches of blue, they help themselves, you get a positive feedback and so on. And so you get the evolution or the stable maintenance of the secretion. Okay, so that seems to work. And indeed, since we published this, a number of labs, including our own, have shown that this works also in empirical systems. But rather than, I normally talk about the published stuff, but I thought I'd give you an unpublished example of this that we're just working on now that shows the same thing. So this is a Pseudomonas aeruginosa project again, and what we're doing here is we're just growing together P001, which is the sort of standard Pseudomonas strain, with and without a plasmid that carries a beta-lactamase. And this beta-lactamase, as many of you will know, will break down penicillin-type antibiotics, okay? And what it does then is it acts as a cooperative trait, because basically in the region around the cell, it breaks down the antibiotic and everyone can grow. And there's the potential then that a guy that doesn't carry the plasmid, which carries a cost, can grow in the shadow of one that makes it. So what can we do then? Well, our prediction, remember from the original cartoon, is if they grow under conditions where there's this spatial structuring occurring, the guy that makes the beta-lactamase should do better than the guy that doesn't, because it's going to preferentially help its own type. And so we can do that. We can grow them in colonies where you get this spatial separation. And indeed, the resistant guy is labeled in yellow in this, and the susceptible in red. And you can see that they indeed do win. Okay, so far so good. It seems consistent. But of course, this isn't really an experimental test. Our experimental test would be, well, okay, if spatial structure is important, then under conditions where they are disordered, then we should expect to see the opposite. And what's very nice about working with colonies, I really like colonies as a system, is that you can just get a pipette tip and each day mix up the colony, right? So each day you just do that, and you do exactly in parallel as in the first experiment. What happens when you do that? Well, lo and behold, the susceptible guy will now win. So just by moving the cells around, you can shift it from a case where antibiotic resistance is favored, a cooperative trait in this case, versus antibiotic susceptibility. So this is quite a nice project just in terms of antibiotic resistance, because we have a condition where under antibiotic treatment, the susceptible cells actually do better than the resistant cells. Okay. So, as I say, most of those ideas are sort of fairly well trodden, so I want to just run through them quickly. But the ideas behind this sort of led us to write a review relatively recently. We called it the genotypic view of interactions, and this is with Sarah Mitri, a former postdoc. And it's captured rather nicely by this cartoon, which was done by a friend of one of my students. And it's basically saying that within a genotype, cells act like a clone, and indeed they become a little bit like the cells in our body. They're a single genetic interest, and so there we can expect there to be a lot of cooperation. But then the question is, what happens, when you look at other genotypes? And the simplest thing is actually that they should be in conflict, but I'll be discussing that more. And indeed, that leads me to the next part of my talk then. What happens when you interact with other genotypes? Well, here again, evolution can offer us some predictions. So, and the key prediction sort of from animal behavior is that there should be very, very strong selection to recognize your own genotype versus other genotypes. So we see this in many different animal groups. I've had, as a PhD student, I had a very personal encounter with this particular mode of behavioral interaction, and it looked a little bit like this. This is the European hornet, Vespa crabro. This is the first species I studied during my PhD. And with a considerable amount of learning, I can tell you that this is their aggressive face. So when you go, and so I didn't take this beautiful photo, but the photographer taking it has actually made them rear up. So when they're rearing up on their front legs, that's bad. It's a good rule for life, I learnt. But basically, this is what they look like on the edge of a colony. If you walk towards a hornet colony, then there's guards on the outside, and they will do this, right? So discriminating me from the rest of the nest is a trivial matter for them, of course. They're actually very sensitive to CO2. So another good rule for life is don't breathe on a honeybee nest. Another one I learnt. And these guys also, though, will do this for foreign workers from other colonies. And they're extremely good at detecting the smell of their colony versus the smell of other colonies. So they have this fairly sophisticated ability to detect the cuticular hydrocarbons, the chemistry of the other workers, and then react in an aggressive or non-aggressive way, depending on where they come from, right? So this is the mode in which I started to study microbes, and I got very interested in this idea, is there this active discrimination? So do microbes actually really sort of detect sort of non-relatives and respond in this sort of behavioral way? And the answer is sort of no in some ways. And it took me a hell of a long time to realize that I was wrong, I would say. But I think recently we've actually worked out how this is going on, I think, in microbes. And it's arguably even more interesting, at least I think so. Yeah, here's our ideas. So what would it look like if a microbe was trying to detect, the strains around it and respond in a sort of, evolutionarily speaking, rational way? Well, we think that one of the key ways they do this is actually through stress responses. And this is work with Dan Cornforth, who's currently working with Marvin Whiteley, who's here and speaking, I think, tomorrow. And so Dan and I started chatting in the corridor one day in Oxford when Dan was there, and we sort of noticed that actually you can take the traditional classifications sort of ecological factors that affect species, so exploitative competition and interference competition, so there are two ways that species interact. And exploitative is basically stealing food, roughly speaking. Interference is being punched or being hit with an antibiotic in the case of microbes. And then abiotic harm. So we noticed those, and then we started to read a lot about stress responses, and we noticed that you could actually sort of line up the stress responses, roughly speaking, to detect each of these types of ecological stress, if you like. So this looked good, but of course it's very easy to make a classification of any sort. You can always line things up. So we really wanted to have a prediction that would test our fit between these things versus an alternative. And our prediction was, right, that microbes tend to use these top two stress responses to detect other strains, but the bottom ones, things like heat, they're detecting abiotic. So what's our prediction? Well, we thought if you get attacked, a very common reaction across the natural world is to attack back. So our prediction then became, well, if you're faced with biotic stress, so these top guys, you should actually be activating your own antibiotics to try and deal with whatever's attacking you. By contrast, if you get sunburnt, right, you may, I guess, get angry with the sun, but you're not going to try and sort of, attack it back. Okay, maybe you will. Not today. So anyway, and so we thought this was an interesting idea, and we spent a long time reading a lot of the literature. This is not terribly systematic set of comparisons, but nevertheless, when we did look at it, what we found was that there's many, many more links where antibiotics are upregulated under biotic stress responses than the ones we categorized as abiotic stresses. This is certainly not a full demonstration of the idea, but it seemed very consistent with it. To the extent that we wrote, a little piece, and it is an opinion piece in Nature Reviews, and we even called this thing competition sensing, as the idea that you're detecting competition by detecting the damage that comes in. But since then, there does seem to be growing evidence that this makes sense. So there's a recent paper by John Michelanos that basically took a reporter of reactive oxygen, put it in E. coli, and then basically attacked E. coli with various things, other bacteria, antibiotics, phage. And in each case could show that actually the oxidative stress response was upregulated whenever it was basically experiencing various forms of biotic stress. But perhaps even nicer examples come from Streptomyces, and there's a couple of papers that come out, but this one comes from this beautiful paper by Matt Traxler and also Roberto Colter, where they grew together different species of Streptomyces and then used a lot of spatial mass spectrometry to understand how that affected the chemistry of the bugs. And this is sort of illustrates it in one slide. Basically, the strain on the right, which is cedar color, I believe, has been put next to another species on the left. And you can see that it's gone bright red in the presence of the other guy. And that bright red is the red pigmented antibiotics of Streptomyces. And so it's upregulating its antibiotics conditional on being right next to this other species. I mean, in this case, we don't know it's a stress response driving it. This has not yet been shown, but we do know that these specific antibiotics are upregulated under stress. But we've also been trying to develop a model system to understand the logic of these things more. And here what we're doing is working with E. coli and the very famous colicins. And colicins are brilliant because of their tractability. And so we've been playing various games, and we're beginning to play various games where we put different strains together and try and understand how they respond to competition from different genotypes. And in this case, the colicins are on a different plasmid, so you can just have two isogenic strains with different plasmids, and they'll unhappily fight with each other. But I just want to show you, this work's just starting, but I just want to show you this sort of illustration of the kind of things that can happen when you start to consider these sort of interactions and responses to competition. So what we have here is, on the left, we have a strain that is making a colicin. It's called E8. But we also have an E8 reporter. So we put a promoter fusion in, which goes green when it's being released. Okay? And so it's green, which means it's making a bit of the colicin. And the guy on the right is just a poor negative strain that we haven't given it any weapons. And what can you see? Well, you can see that basically there's some inhibition between the two strains. So the guy making the colicin is inhibiting the strain on the right. I mean, I say that poor guy is poor negative, but what's interesting is what happens if we arm that guy. So now what we're going to do is take that negative strain and give it its own colicin, right? So we're going to give it, this is sort of with a nod to Marvin the Texan sitting at the back. What we're doing is we're basically, this is the argument in Texas that if someone has a gun, you should always give the other guy a gun, right? Okay, so let's see how that works for them. So this is what happens then when we give E2 the colicin. What's happened? It's worse off. Why is it worse off? Because it's making a DNA-damaging colicin. Colicins are upregulated, conditional on DNA damage. And so what it's actually doing is it's inducing the guy E8 on the left to actually make more of its colicin, and it ends up worse off. So this is sort of mutually assured destruction. So this is a very simple system where I mean, we're studying it specifically because we understand nearly all the mechanisms, although we are finding out new things on the way. But you can see, how simple, very simple rules, such as upregulating based on attack, can lead to rather interesting outcomes that indeed have analogies from other social systems, if we can call Texas a social system. Sorry, Marvin. It's too easy. Okay. I did live in Texas for 3 years, so I'm speaking with a certain amount of well-earned prejudice. Okay. So I'd like to then, at this point, sort of pause a moment and then sort of come back to the title of the symposium, because I've been thinking a lot about the title of it and the use of the word of communication. And communication is used in many ways, and so I certainly don't want to say that we can't classify what I've been talking about as communication, although evolutionary biologists would not normally call it so. But what I have been showing, right, is a series of cases where you take two strains and you put them together and they interact and affect each other in a way that strongly affects their phenotypes. I mean, I've chosen cases where we really knew what was going on ahead of time, but nevertheless you can see that you put those two E. coli strains together, suddenly something has happened, something has changed. And I guess what I would say is, I wouldn't call that communication, at least not the examples I've shown. What I'm seeing is strong effects of one strain on the other, but in this case it's actually sort of detecting attacks and responding in kind. And so when we bring like a word like communication into microbiology, I think it's very important that we think about whether it is just the kind of things I'm showing, or whether it really is an evolved system where two guys are trying to communicate actively for their mutual benefit. And that's, I think, an interesting question to think about throughout the symposium. But I'd also like to sort of show you a test case of this kind of idea from a project that sort of started a long time ago, but also, as you'll see, actually ends up with a very similar conclusion. So this project was actually when I was really I was hoping to discover a way that microbes detected, each other and responded, right? And so back in the day when I was at Harvard, this started about 10 years ago, this project. I basically took strains of Pseudomonas aeruginosa, and you can see it was just published like last year, so this was a long journey. Arguably the most inefficient science I've done, but hopefully quite interesting nevertheless. So all I did was take different strains of Pseudomonas aeruginosa, which, and like proper natural isolates now, so things were isolated actually from different places and different patients, and just mixed them up together and did one of these classic biofilm assays. So the bottom left, for those of you that have never seen these things, I've seen far too many of them, this is a crystal violet assay. And all you do is you grow cells in a microtiter plate, the cells attach around the surface of the liquid, and then you can basically wash out everything except the attached cells, and then dye and solubilize things. So you get basically a signal of how much biofilm you had, how many attached cells you had. Okay, so I did that very simple assay, and then we added different numbers of strains into the culture. And we calculated the biofilm index, which is the ratio of cells in the plankton to the cells that were attached in this assay. And we saw the following: more strains, more biofilm. So that seemed interesting. I mean, it kind of went against our prediction. We thought it was going to be all-out war and everything going wrong, but we actually saw more biofilm. Okay, so far so good. That's a very crude assay. Well, let's do a slightly less crude one. It's still these microtiter assays. But now let's look at it in a fairly defined way. So here we're taking strain 1 and 4 in this case, and 4 and 69, and then we're changing the proportions of them, and the blue guys are the biofilm, and the red are the cells in the planktonic phase. Okay? And so you're seeing the same thing, just recapitulated in a dose-dependent way for two strains. When you mix them, you basically get more biofilm and a little bit less plankton than when they're on their own. Okay. So again, this started to look maybe like they're working together to do something, right? But then that sort of illusion shattered fairly quickly when you start to actually label the strains and look at the biofilm. And basically, it's nearly only one of the two strains that actually persists. So you end up with a case where there's more biofilm, but one guy is also eliminating the other during the process. And I can show that more sort of starkly here. These are 5 different strains, which we mixed up in all combinations. And down the sort of middle diagonal are the controls, where there's 50-50. But every time you move away from the diagonal, you basically end up with almost complete elimination of one of the two strains in the biofilm. Okay. So it's an intense competitive environment, but we are getting more biofilm. So what's going on? Well, the first thing we wanted to do was actually move away from these crystal violet assays. Anyone that's done them knows that you can easily convince yourselves of things that aren't true with them. They're a slightly messy assay, depending on how long you do things and exactly, you know, the way you measure things. You know, you can start to worry about it. So what we did was we went to flow cells and wanted to just do the same experiment and convince ourselves we do see the same phenotype, and indeed we do. So all we're going to do now is mix our two strains, put them in a flow cell, and then run the flow cell. And I'll just show you a quick movie of this. So I'm only showing you the yellow cells here because our CFP signal isn't terribly good in this scope. But you can still see the effect nevertheless. On the left, we've got 7 with itself. On the right, we've got 7 with another strain, which is not image, but you can basically see you get these much bigger biofilms. Okay. So what we're seeing is elimination and biofilm formation in the presence of another strain. So what's going on? What drives this response? And actually, many of you may well have guessed, it's basically antibiotics. Well, it's not strictly antibiotics. In this case, it's these guys. And so these are these cool phage tails. They're called pyacins, R-pyacins in Pseudomonas. And these are modified phage tails. They don't have the capsule anymore, or capsid rather. They have only the tail, and basically they're basically a brutal puncturing device. So what they do is they attach to non-self strains, so they have a way of not attaching to the LPS of a self strain. They attach them and just punch holes. Physically, they're hole punches. Okay. So these guys cause a widespread cell damage, and indeed we can show that actually they're predominantly responsible for our response. So how do we do that? Well, we did various things, but here's one key experiment. So we're using these nice flow cells where you can flow in 2 different things and create a gradient of them. So the flow is going from right to left here. We're using TB, which is the typical biofilm medium that's been used with Pseudomonas for a long time. And then we can put in another factor on one side and ask how this affects our system. So the control is just you add TB, you let things grow. But then what we can do is we can take the spent media of another strain. So you grow up the cells, you take out the cells, you take the spent media, and then you flow that in along with the media. What happens? Well, first of all, you get a killing zone because it is inhibitory, but you can see the moment you start to get to lower concentrations, you get this strong induction of the biofilm response. Okay, so we can isolate it to this. Then, I mean, this was not entire guesswork. We did some chemistry and fractionation and such, but basically what we can do then is just knock out the regulator of these pyacins, or indeed the specific loci for the R-pyacins, and do the same experiment. And if you do that, you know, you don't see the response. So you can really get this strong induction of biofilm formation just by having cell-damaging agents in the media. And in this case, it was R-pyacins, but actually our intuition with the other strains is it may be other negative factors that drive this. And indeed, we can ask the question, right, how general is this response? Anyone that studied sublethal antibiotics, I imagine there may be people here, will actually know, and I should have picked up on this much earlier myself, that sublethal antibiotics induce biofilm formation in a whole load of species, gram-negatives, gram-positives, under all kinds of conditions. There's evidence that sublethal antibiotics make cells make more biofilm. And indeed, if we do those we recapitulate those experiments in our device, so putting in various antibiotics with different modes of action, we get very similar phenotypes to that which we saw when we flowed in the spent media or another strain. So these are, you know, clinical antibiotics, and you get, again, this very strong biofilm induction under exactly the same conditions. So our idea then is that this is actually just a general response to cell damage. We don't, of course, know, and it may be reactive oxygen. We don't know if there is indeed a common pathway driving it, but what it does suggest is that these attacks that bacteria induce on each other have clinically relevant phenotypes that are manifest, you know, when you start hitting them with antibiotics. It also goes back to this question of communication, because as many of you will know, you know, there's been this theme in the literature in microbiology that maybe antibiotics are actually signals, you know, so that they're at low concentration in the environment, and that as a result of that, and also the key thing is that people often report very strong physiological responses to sublethal antibiotics. And those responses have been interpreted as communication, as signaling and cooperation between microbes. And I think these kind of data strongly caution against that. I do think there's really, really strong responses to sublethal antibiotics, but I think they occur not because 2 guys are trying to communicate with each other in some cooperative way, but it's basically because it's warning of an impending attack. Right? It makes a lot of sense. If you start detecting sublethal antibiotics, that could easily be a prediction of lethal antibiotics. So you want to get yourself going. You want to basically make biofilm, which we know defends you against antibiotics, but you also probably want to respond in kind and start upregulating your own attacks. And indeed, this idea of, tit-for-tat attacks is well studied now in type 6 secretion, and Melanie will be talking about that as well here. Okay. How am I doing for time? Okay. So this returns me then to this sort of genotypic view then. And so I would say, the first part of my talk, I was talking about this idea that within a clonal group, we expect a lot of cooperation, and that's where I expect to see the real communication happening most of the time. But beyond that, you basically have these other genotypes, and often those genotypes are using your resources, and also, as we know, they've often evolved antibiotics to attack you. And so there, I think, a lot of the interactions that are going on are competitive, and also based on this idea of detecting attacks and defending and responding in kind. Okay. But for the last 10 minutes, I'd like to just open it up a bit further and ask what happens when we consider another individual. And this is my chosen individual. It's obviously a host. I like polar bears, so I chose a polar bear. But the question then is, what happens, and I think this is a particularly fascinating question for an evolutionary biologist, what happens when we take these societies and all these interactions and then put them inside of another organism that itself is evolving? How does that work? If you could take a rainforest and put it inside another organism, that suddenly, would that change the rules of interaction? Would that change the way the ecology of the system behaves? We can't do it with the rainforest, but in fact, this has happened, over hundreds of millions of years with the evolution of microbiota. Microbiota is just another microbial community, but one that's inside an evolving host. And so then that leads to this sort of fascinating question, which is what happens inside a host to the interactions within the microbes. So when I talk about these things, I'm often imagining something like this, and this is sort of this beautiful cross-section of a mouse, a healthy mouse, at least at the time that the section was made. And this shows the epithelia on the bottom left, the thick mucus layer, which is that sort of clear area, and then the sort of thin, looser mucus layer, where you have these dense populations of microbes. So we know we have these dense populations of microbes growing in there, and the question is, what are they doing? What are the rules inside them, inside those communities? I've talked very much about this idea that, you could have either very competitive communities where things are interacting negatively, or maybe you have cooperative communities where things act positively. Well, we've come down pretty hard, I didn't talk about this all that much, on the fact that both from theory and from experiments, that we expect a lot of the interactions in microbial communities to be of a competitive nature. However, if you read the microbiome literature, people often talk about its cooperative nature, not only with the host, but also within the communities. And there's a sense behind that, right? It makes sense that a host would want to have these productive, harmonious sort of societies of microbes inside it that are sort of producing things for it. At least that's sort of the metaphor that's often used. But we wanted to just kind of dissect that metaphor and ask, does it actually make sense for a host to have these cooperative communities? And so this is work with Kat and Jonas. Both the former students are now working with Joao at Sloan Kettering. And so these guys just started to analyze this problem using sort of classic population models from Locke of Volterra, and also made famous by people like May. And these capture these complex networks of interacting species in terms of a few key parameters, including how things interact with each other and how a species interacts with itself. So we can do this, and then we can say, okay, what does the community look like if I then just dial up and dial down the amount of positive interactions among my species and my network? So, and on the left we have a competitive, and on the right we have cooperative. Well, if you do a competitive system, you have a set of species that exist, and then you kick them. Well, they'll bounce back. Okay, so they're rather stable. I mean, here this is in a simulation, so they're sort of fluctuating around, but they actually make this nice stable system. But what's interesting is what happens if you do the same thing with a cooperative group. So now, if you have these coexisting species and you punch them, then basically they'll very often just sort of spiral out of control and collapse. So these cooperative systems turn out to be very unstable. And there's a very simple intuitive reason for this that anyone who does network theory will well appreciate. And that is that if you have cooperation, so plus-plus interactions, you create mutual dependency. So now I've got two guys that are helping each other, and I pull one down. This one needed the other one, and it gets pulled down with it. And so it actually makes a very fragile system. By contrast, if you have negative interactions, you end up with negative feedback loops. And we all know from cell biology that this is basically a stabilizing process. And so we can analyze this much more generally. We did it using various mathematical and computational techniques. But basically, in each case, what you're seeing on the bottom is the proportion of cooperation, PM, and then the stability of these systems, measured, as I say, with just maths, with permanence, which is a computational technique, and then individual-based modeling, which is really sort of simulations. Basically, in all cases, things basically collapse if they have too many positive links. So this leads to a prediction, then, that we expect non-cooperative and weak interactions will be key to actually having a stable microbiome, even if that is less productive than having all these cooperative interactions that are working for mutual gain. The question, then, is what actually happens in the microbiome, and we really don't know that much yet. But we were very lucky that a few years before us, João had published this paper where he'd gone to incredible effort to try and estimate the sort of interactions that occur at the genus level, admittedly, between various microbes inside a mouse that was hit with an antibiotic perturbation. It was the perfect test of our model before we'd actually done our model. And so in that sense, we were very lucky. And what does it show? Well, what it shows, first of all, on the left, is that the interaction strength between species normalized relative to 1, which is the interaction with yourself, tends to be low. So they tend to interact fairly weakly. That's one of our predictions. The other thing is that the proportion of interactions tends to be either of the competitive or exploitative type. They tend to be either cases where you are negative-negative or plus-negative, so someone taking something from you. And there's a very small proportion of cooperative ones. And so, as we predicted then, you do tend to see non-cooperative and weak interactions dominate in this first analysis. More work needs to be done here, but nevertheless, it was very encouraging that it seems to fit with our simple predictions. Okay. But there is a plus there, right? And so that then begs the question, how often do we see cooperative interactions in the microbiome? And indeed, are they important? And in a parallel project that I've been working on with what you'll see who it is in a minute, Seth, then we've basically been talking for a long time is actually really cooperation in the microbiome. And so what do I mean by cooperation? Let's just make that really clear, because I'm talking about cooperation between species now. I'm not just talking about, the idea that you secrete something that helps your clonmate. I'm talking, sort of imagery like this. This is a photo I took on my holiday. This is from this is where my entomologist side is out nowadays, taking photos of insects. And this is a classic plant-pollinator relationship, right? And here what we have is the plant has evolved nectar to feed pollinators, and the pollinators in return are moving pollen around, right? So this is an evolved example of cooperation. And what we wanted to ask was, do we see things like this going on in microbial communities, but particularly inside the gut? And so where would you look for that? Where would you find something as beautiful as this inside the gut? Well, here it is, its own beauty. Arguably not quite as charismatic, but of course we know that the Bacteroides, which are these, the major group of bacteria inside our guts, spend a lot of their time and energy breaking down complex carbohydrates. It's why we like them, in quotes, because they do seem to do a lot of good in this way, and also generally high Bacteroides titers tends to be good for our health, so they're a well-studied group. And what we know is that they break down complex carbohydrates outside the cell. And in particular, it's this top bit I'd like to focus on. They have all these enzymes, often with this sus designation, which are breaking down things like inulin and other complex carbohydrates and then pulling the monomers inside the cell. And so when I started talking to, this was his Seth, and it's also worked with Laurie Comstock, who are at Harvard Med School. We thought this is a good place to look for cooperation, either within or between genotypes. And we did various things with this, but I'm just going to tell you about the most interesting one. So in this case, we took Bacteroides ovatus, okay, and it has this enzyme. In fact, it's two enzymes that sit outside its cell that break down inulin. Okay, we knew all that ahead of time. And what you can do is you can grow this guy on inulin. Okay, there's a bit of a detail here. In the absence of the monomers or monosaccharides, they don't actually activate the system. Okay, so you don't turn it on, but hopefully on the right-hand side of that, don't have a pointer, but anyway, the bottom right, we've got inulin growing with either glucose, fructose, or basically fructose oligosaccharides. And you can see when you look up that they grow well so long they have a bit of the monomer and they have the inulin. Okay, so they can grow on inulin. So far, so good. So then what we asked was, what happens if we delete these enzymes that are important for the breakdown of inulin? What's the prediction? If you think about it, the prediction is they use this to break down inulin. If you knock out this locus, they shouldn't grow anymore, right? That's what we expected. The crazy thing is they grow better when you knock out these enzymes. So again, if you focus on the thick bars, which is the conditions under which the enzymes are getting activated, they're growing better in each case when they don't have the enzymes than when they do. Okay, so it's all a bit weird. So what's going on? Why are they growing better without enzymes that they need to break down inulin? And so this to us, I mean, we spent a long time trying to work out what was going on, and this is done in liquid, so we thought it's maybe cross-feeding within colonies, and we did all kinds of different experiments. But in the end, basically, we started to convince ourselves that this looks like really like costly cooperation. What this started to look like to us is the plant making the nectar. It's a bacteria that's breaking down this sugar, but actually it doesn't need to break it down. In fact, we could subsequently show they can just take inulin in without breaking it down. They don't even need to break it down, and the reason for that apparent cost is just that when they don't break it down, then basically they can eat it much better. So, how do we answer this? Answer this sort of strange phenotype then? Well, you can see where I'm going. Well, you just have to consider another species. And we did this in vitro, but also in vivo, and I'll show you the in vivo experiments. So this is now giving mice basically the two strains. We've got our wild type, which has the two enzymes, and the mutant that lacks both of them. And we're putting them on a polysaccharide-free diet in the first instance, and you can see basically they more or less stayed the same. In fact, as you would expect from the previous experiments, the mutant does slightly better. Okay, then we can give them inulin, which is what the enzyme actually will break down. And again, we see no effect. But what's interesting, what we can do then is add in Bacteries vulgaris, which is a common species that occurs with ovatus in the gut, and one we know can use inulin breakdown products. And now, suddenly, the wild type does much better. And we also did these experiments in colonies, and you can show that conditional on the presence of vulgaris, there's a benefit to breaking down inulin. So what's happening is that well, here's a little cartoon. We have a case where ovatus is breaking down inulin, which it would prefer to eat directly. That's cross-feeding vulgaris, and then vulgaris is helping them back. We don't know how they're helping them back. We've started to play around with that, but we haven't got it yet. But nevertheless, it really looks like evolved cooperation. And the analogy is pretty strong to my original plant pollinator, perhaps more than I realized when we started this, because they really are producing, a sugar, which is feeding another guy that's helping them back. But obviously, the feedback benefit is going to be very different, and we need to work that out still. Okay, I'm on time. So just quickly then to summarize what I sort of said today. So what we've done sort of really suggests that cooperation within genotypes is very common. And indeed, so long as we can get these patches of a single genotype, that's where we really expect to see a lot of cooperation. By contrast, between different strains, we think that nutrient competition will commonly lead to competition. And indeed, I argue that even that things like stress responses have evolved, at least in part, to detect and respond to this important factor that every microbe has to deal with, which is competition with other genotypes. And I also argued in the context of the symposium that maybe a lot of what people might call communication is actually just detecting attacks and responding in kind. But I think that's an interesting question for discussion. And then finally, you can ask, well, what happens when we put these things inside a host? Well, again, we see competition, I think, dominating, as would be expected under basic evolutionary rules. But also, I argue that may even be good for a host because it can create these stable systems. Nevertheless, it was a long journey, but we did also, I think, find evidence for evolved cooperation between two species that, arguably is even as exquisite as the ones we see at the macroscopic scale. So that we need to also consider the possibilities of cooperation in these systems. Indeed, that they may be very important. In that last case, ovatus and vulgaris, can make up several % of your total microbiome. So this is a major interaction and no doubt has important health effects. And very finally, I'd like to acknowledge several of the people that did this work, including, as I say, Jonas and Kat, who are now working with Joelle. And thank you very much for the opportunity to speak and for your listening.